Okay, Drosophila Transgenics. So Drosophila is a very important model organism, obviously, especially for transgenics. Drosophila was probably the fastest and most adept model at which people did transgenic insertions into animals. So historically, this is an important model, and there are many advantages to it. And many of the tactics that are used in Drosophila for molecular insertion of transgenes are also adapted, or similar methods are used in other organisms. So it's kind of an important model. Okay, so there's been sort of like different phases of ways to do transgenics in Drosophila. So I'm going to go through the history of the different phases. So the first phase was using P elements. Okay, so the P element is a key term. So P element is a Drosophila specific transposon. So this is a transposon, a jumping gene that is able to be used to insert transgenes into Drosophila. This was originally discovered from this phenotype called Hybrid Dysgenesis. Try to spell it right. Okay, so I'll talk about Hybrid Dysgenesis. Hybrid dysgenesis is this kind of bizarre phenotype where if you have a male, so these are fruit flies, okay? So if you have a male fruit fly that is P positive, so it's got a P element inside of it, and if that male crosses with a female that's, let's say, P minus, actually the technical term they use here is M, but we'll just say minus. So there's P positive male. If he crosses to a female that does not have P elements, these embryos die. Okay. So first question that you should contemplate or think about is why might you get embryonic death in the case of this cross? What do you think could be a molecular cause of the death? So remember, a P element is what?
A transposon.
It's a transposon. So there's a case where a transposon is killing an embryo, in this case. How could you get death, embryonic death from a transposon?
It just goes somewhere and interrupts something to make it lethal?
Yes, that's pretty much precisely what happens. So in the case of the sperm that has P elements inside of it, so in that sperm are chromosomes, and in those chromosomes, let's say, is the P element, okay? When that sperm combines with an egg, in this case that does not have the P elements, as soon as this chromosome enters the zygote phase, okay, the P element starts expressing. And if transposons go expression crazy, they get expressed a whole bunch, and then if they're expressed a whole bunch, they're gonna start jumping around, okay? So they'll just start jumping. And if they're jumping around in an early embryo, essentially what's happening is it's just shotgunning the genome with insertions of transposons completely randomly. So that is how you can get embryonic death, is it's essentially a shotgun at the genome of an early embryo. And if any of those transposons insert into a key essential gene, this embryo is just gonna completely die, okay? So that is the phenotype of hybrid dysgenesis. That is how they originally discovered P elements, is they had this weird phenotype, and they traced the source of the phenotype to the gene that was encoding the P element. Okay, so that's half of the story of the P element. The other half of this, it gets a little bit complex, but it's interesting and it's worth talking about because we'll learn more about this stuff later. So the other side of hybrid dysgenesis is if you have a P element, positive male, and he crosses to a P element positive female, these embryos survive. So this is kind of like a bizarre molecular rationale. Does anybody have any hypotheses why this one would die, but this cross would survive? So in this case, the only difference between this cross and this cross is that the female has the P elements as well. generate some hypotheses about what could be happening. How might these ones survive, but these ones would die? This is very counterintuitive.
I mean, it jumps less for some reason. I just don't know why it would.
Okay, that's very good, very close. So there's something that's causing it to jump less. That's true. So there's something in the female that's causing it to jump less. And we just talked last lecture about unique phenotypes that can come from the mother's genome. Remember what that term was, those types of genes?
Maternal effect.
Yeah, so this is actually an example of a maternal effect. Okay, so females that have the active P element in their ovaries and in the nurse cells and in the eggs that get produced by these females, they actually overexpress a silencing system called PIWI. So I haven't yet taught you guys how silencing works. I will teach that later in the RNAi stuff. So it's a mechanism of RNA interference. Just essentially know now that there are systems, there are genetic systems that have evolved that can shut genes off, and that happens through RNA interference. We'll talk about those mechanisms later. But this female actually has, if she has P elements, she would die unless she is constantly expressing this silencing system. So it's like she has evolved a resistance mechanism against the P element. And so any future offspring from her are inheriting this PIWI silencing system being expressed as a maternal effect gene. So the silencing of the P element is actually a maternal effect, which cannot be conveyed in this situation, because in this situation, she hasn't been exposed to the P elements and she's not expressing the silencing system. So that's actually an example of a maternal effect gene phenotype that comes from the mom and saves the embryo in this case. So this is the case of hybrid dysgenesis. And again, this is the biological case that caused the discovery of P elements.
Sorry if I just didn't catch it, but why would the female die and not the male if she had
The male that's P positive, where does he come from?
Where does he come from?
Where does he come from?
A female that's...
A female is P positive.
Mm-hmm.
So all the P positive survivors source back to a female that was a P positive that was expressing the silencing gene. Does that answer your question?
Okay. Yes.
So that's how you can get males that are still
Yeah.
alive, is because they came from moms that had
Yeah.
the silencing expression system expressed. So it's only in the case where you're crossing to a completely naive female
Yeah.
that's P minus, has never been exposed to P elements, they're not in the genome, that's when you get the death. And the reason people discovered this is because Drosophila is such a great model that people had collected fruit flies from all over different parts of the world, and some of the fruit flies had the P element, and other populations of fruit flies did not have the P element. And because crossing is very common in Drosophila, so if you're crossing different strains, you'll find these weird, bizarre phenotypes. Did that kind of answer your question?
Yes, it did.
Okay, so that's hybrid dysgenesis. Okay, so let's draw a classical P element vector. So P elements, then once they kind of discovered that there is this inherently very hyperactive jumping transposon, which is active in the germlines, which is nice, in Drosophila, they immediately thought, okay, well, if this is a transposon that's active in the germlines, that could be a great tool for biotechnology, because if you could get it to jump properly in early germlines, you could get insertions into the chromosomes. Okay, so they tried to engineer the system. So this should be a review again. In classical transposon systems, there's the helper and the donor, okay? And so in the case of the helper-donor system for Drosophila, they have a helper plasmid which encodes the P transposase. So something like this, that's the helper. And transposases recognize what types of sites. So transposase is actually a gene that codes for a protein that binds to DNA, and it binds to what type of sites? Does anybody remember what they're called? Otherwise, this is a good review, I guess.
Transposases?
Okay, so we know that transposases are jumping genes. they get transcribed into messenger RNA, that gets translated into a protein, that protein comes and it binds to DNA and it grabs stuff and it cuts it out. What's the site name that it binds to?
Landing site?
We'll talk about landing sites. You might call it a landing site. It's actually called an inverted repeat, right? We remember this? Inverted repeats are just, this sequence is going this way and it's the same sequence flipped going the other way. That's an inverted repeat. So it's a repeat that's flipped in directions. Transposases recognize those. So again, this should be review. So you can put your gene X in here. And if you do this, this transposon, the Drosophila transposon, eukaryotic transposon, you grab this and pop it out. Some of the differences in selection, we have already talked about this, so again, this should be review. The selectable marker in Drosophila, the most common one is called mini white, okay? Or you'll sometimes see just W. White is a gene that codes for red eyes, okay? So in a normal Drosophila, this is wild type. Oh my God. The time when I need the red marker. Okay, in a normal Drosophila, this is wild type. So in a normal Drosophila, they have red eyes, okay? That means they have an intact copy of the white gene. When you do transgenics in Drosophila, you are never working with wild type. This is just kind of like yeast in a similar way that the yeast that we work with in the lab are not wild type yeast. They are genetically modified yeast. The Drosophila that we're working with transgenics in the lab are not wild type Drosophila. They are most commonly the white minus phenotype, okay? So that means that their native copy of the white gene is deleted or disrupted or damaged. And so the Drosophila that we work with in the lab have white eyes, okay? So then if this gene inserts into this line of Drosophila, its offspring will convert to red eyes, and then you know it has a transgene. So the way that the transgenics work is when they do these transgene insertions, they'll build their vector, their donor and their helper. They will mini prep these. They will put them into a microinjection needle. Okay, so these get loaded in the needle. They have a W minus, you might call it strain, okay, of Drosophila that has white eyes. They culture these, just like you guys are culturing Drosophila right now. So you'll actually learn precisely, if you have not already, how we set these up so that they lay a bunch of eggs. They set them up in an apple juice plate so that they lay a ton of eggs, okay? And this is actually a really important thing for biotechnology and for transgenics in animals. The one advantage that Drosophila, there's many advantages that Drosophila has for biotech, but one advantage is you can get synchronous egg laying, where you can get Drosophila to lay hundreds and hundreds of eggs right when you want them to, and all you need to do is you just add a mixture of yeast and water. So you make this like peanut buttery yeast, you put it on an apple juice plate, and we will do this in the lab. So you have a little smudge of yeast, you put a bunch of flies in there, the W minus strain, they will lay a ton of eggs, okay? Like right when it takes a little bit of time for them to kind of like ramp up egg laying, but then they'll start laying eggs. And then once they're laying eggs, somebody will take these eggs and collect them with a paintbrush and assemble them onto a slide in a row like this, okay? So if you can imagine, there's a slide that just has a bunch of Drosophila eggs, W minus strain, what would you call it? Stuck to it. And then they just take the needle, companies do this, they take the needle and they just go all the way down. And so they just inject all these eggs with the helper and the donor plasmid. And this is all before what key stage of development?
When they all the nuclei get their own cells.
Yes, when all the nuclei cellularize, the term for that starts with a B. That's, there you go, that's the blastoderm. So before blastoderm formation. So typically in Drosophila, when you're microinjecting, you're typically microinjecting with eggs that are less than 2 hours old. As long as you're microinjecting with eggs that are less than 2 hours old, you're probably pretty much before the blastoderm formation. So they're injecting these, they inject them, and after they inject them, they're gonna get some kind of what's called a hatch rate. Okay, so you get a hatch rate. So this is a very traumatizing and damaging process when the needle is poking the egg. So if you get really, really good at this, like the professional companies, they can get pretty much like a 50% hatch rate. So that means of all the eggs they inject, 50% of them just outright die from the needle, the damage that the needle causes. But the other 50% will survive, okay? So out of this, say if you injected 100, you'd get maybe like 50 survivors that turn to larvae, okay? And then they rear the larvae through the pupal stage to the adult stage. And typically what comes out then is most of the flies that will come out will be white-eyed, but amidst the population, there will be a few individuals that have red eyes, okay? So then they know in this population, this one's the transgenic, right? This one is the one that actually received the transposon insertion, okay? And then they take that individual, erase all this now. They take that individual, and what's the problem or the conundrum with that individual at this stage? What are you worried about at this stage? It definitely has a transgene, but what could be the problem? So imagine you injected an egg that was 1.5 hours old. So blastoderm had not formed yet, but you had a bunch of nuclei, okay? So this is a pretty already, it's 1.5 hours already developed. There's a lot of nuclei. You injected those two plasmids, the transposon does the insertions. What do you worry about? It generates this fly. What do you worry about?
Whether or not all the cells adopted it?
Yes, that's what you're worrying about. So the question is this a chimeric? That's one question. Is this a chimeric? Do all the cells have the insertion or not? That's the first question. The second question is, there's really only one cell type that you care about that must have the insertion, which is the what type of cells? Actually, it's not even helpful if the eyes have the insertion, because why is it not helpful if the eyes have the insertion? Well, I mean, in some sense you'll see, yeah, that's how it generates the marker. But if the insertion is only in the eyes, what's the problem?
Oh, then it appears transgenic, it's actually not.
Well, it is transgenic because it has red eyes in this case. So it's still transgenic, but what's the problem if the transgene is only in the eye cells?
It doesn't have the thing that, like Gene X, right?
No, it does.
Would it just not be able to pass it down?
Yeah, it's not inheritable if it's in the eyes. So it has to be in what type of cell?
The sex cells?
Yeah, the germline. So these are the two questions you worry about. Is this fly a chimeric? And if it is a chimeric, you wonder, is it actually the germline cells that got modified or not? You don't know. You don't know at this point. So how would you figure out if the germline has it? What do you need to do? What's the experiment you need to do?
You just have to do cross.
Yeah, you breed it, right? So then what they do is they take the red eye. They take the red eye and they'll cross it to another white eye, female, or depending on what the sex was, like I guess it wouldn't matter, you could cross either male or female. But they'll do this cross, okay? And you know it was in the germline if what happens?
If you get the red eyes.
It's not gonna be all red eyes, right? Because there's classical Mendelian genetics. But if you got, in this case, 1/4%, so 25% offspring, if 25% of the offspring had the red eyes, then you know you hit the germline. If you got all white eyes, that transgene was, or that transgenic insertion was garbage because it didn't hit the germline. So you just throw that line out if you got all white eyes. But if you got some red eyes, 25%, that means you hit the germline. So then what do you need to do? So in this case, if 25%, that means the transgene is not homozygous, it's heterozygous. So remember, Drosophila are diploid. So if this, the genotype of this thing would be transgenic X over wild type. So this is one copy and a second copy. One copy has the transgene, one copy does not have the transgene. This case is wild type over wild type. That's how when you get the cross, you get 25%. So you don't really want to keep transgenes as this genotype because this can be unstable. So you want to convert this to a homozygous, ideally you want to convert this to a homozygous transgenic over transgenic. So how do you generate that?
You breed it with another red eye?
Yes, so if you have 25%, very good, you have 25% of the flies have red eyes, then you just take another one of these from that same generation, and you do an incestuous cross with its brother or sister, and that will give you, let's see if it's 1/4. This will give you 50% of the population will then be transgene over transgene. Okay. What?
Did I word that wrong when I was saying to breed them?
Did you read that wrong?
No, I said, did I word it wrong?
No, that's correct. Breed them.
Yeah, but earlier, like, you started explaining about like the different like wild types. Right? You asked like the question, and I was like, you breed them. But should I have been more specific? Should I said you like should have bred both heterozygotes?
Well, I guess that's more the. Well, I don't know what you breed the red eyes.
Yeah.
That's all I'm saying is you take the red eyes, you keep crossing them to each other, you keep breeding them to each other, and if you keep crossing red eyes for red eyes, eventually your entire population will look like this. And when your entire population looks like this, how would you know phenotypically?
You only ever get red eyes.
You would only ever get red eyes. So if there's no longer white eyes ever in the population, then you have a homozygous transgene, and you have successfully like constructed your transgenic Drosophila.
And you need to first breed it to the white eye. Like, it's important to do that step to make sure that it
Yes.
tests that.
Yeah. So the first, I mean, in theory, you could breed. These are called the ones that actually are injected. Those are called the F0s. You could breed a red eye F0 with a red eye F0. You could. That might speed up the process a little bit. But to standardize the process, they kind of always cross it to. It's called an outcross to a white-eyed line. But in general, what you're doing is you're breeding the red eyes. Okay. Then once you bred all the red eyes, you got your transgene insertion. Okay, we did that. Okay. How much time do I have left?
25 minutes.
Okay, good. Okay, so this works really well, and this was very successful. And microinjecting is manual labor and it's painstaking, but it's actually not that difficult for Drosophila. So this sort of mass-enabled transgenic insertions. The first thing that people would mostly use this for, even before they were inserting transgenes, was reverse genetics. So again, this should be reviewed. We understand what's the goal of reverse genetics?
Cut stuff out to trace.
So you said cut stuff out. In this case, cut stuff out is not correct. Why is cut stuff out not correct?
Because that implies.
Nothing's getting cut out here. Well, at least in the case of using P elements for reverse genetics. What's a more accurate way to say what's happening instead of cut stuff out? What's happening if you use P elements for reverse genetics?
It's moving, like,
Oh, P elements specifically.
Yeah.
Okay.
It's a random insertion.
It's a random insertion. So these are insertions. And a random insertion is going to randomly break some gene. So this sort of like enabled mass reverse genetic experiments in Drosophila that they could trace back to where the insertion was. So that you could always do reverse genetics with any organism if you treat it with like chemical mutagens. But the problem with mutagenizing and doing reverse genetics is it's very difficult to detect the site of the mutation. It's very difficult to do that. In this case, it's very easy to detect the site of the insertion because you know exactly what is getting inserted. It's the typically it's the P element transposase. So if you have the P element transposase with its own inverted repeats, this is what gets inserted into the Drosophila genome. And so if it got inserted right in the context of some gene X, that can often then produce a phenotype. So imagine, as an example, we inserted into some Hox gene X, which controls the wing, and you got some weird, bizarre wing phenotype. You could figure out the gene that was controlling that wing phenotype. How could you do an experiment that would actually identify the site of the insertion? What could you use to figure out where the insertion happened? So you're looking for this, and you're trying to figure out where it got inserted.
Did you use a GFP tag?
So a GFP tag is going to be used for seeing localization of a protein in a cell. So that's not going to tell you the localization of the insert in a genome.
Yeah.
Does that make sense? So just to clarify, what type of molecule are we working with here? DNA. So we need a way to find a site in DNA. What could we use?
Maybe live cells?
You don't even need cells, period. You just need an extraction of the genomic DNA, and then you could do things with that genomic DNA to figure out the insertion site. What are some techniques that we could use? What techniques can be used for DNA to find things like this?
Sequencing.
Okay, we could do sequencing. So we could just sequence the entire genome, and then we would do like a blast analysis. We would look for this, and the assembly would tell us where it was. That would be kind of like a very expensive way to do it. What would be the cheapest way to do it?
Like fish?
Fish would tell you what chromosome. So like if you did fish for this probe, you'd essentially, Drosophila have chromosome 2, 3 dot chromosome, and then XY chromosomes. So if you did fish, you maybe would see something like this, and then you could conclude, okay, the insertion site is on this arm of chromosome 2.
Would you just do a PCR?
Yes, you could just do a PCR. So you could design a PCR primer that was inside of your, so one PCR primer, maybe your forward primer, is inside of your insertion, your P element. And then you can do what's called like degenerate PCR, which is you have kind of like crappy primers that are kind of just bind sloppy. So if you have a degenerate PCR primer that's kind of just sloppy, it'll find a binding site somewhere up here, and then you can amplify this product, and then you can find the insertion site by saying you're sequencing this PCR product, and the sequence that's right here will tell you where the insertion site is. Does that make sense? So you can actually, this is, what I'm saying here is this was an amazing technology because it enabled really advanced high throughput reverse genetics where you could just immediately find the site of insertion with a simple like PCR reaction. And so you could trace the source of the insert, which would trace back the phenotype, okay? So that is how they could quickly do reverse genetics using P elements. Okay, flaws of the P element system. It's great for reverse genetics. It's great for inserting transgenes, but it's random. Okay, that was a negative thing. So later, so that was the first generation. Second generation of Drosophila transgenics sort of fixed the randomness problem. Okay, so the second generation of Drosophila transgenics relies on what's called PhiC31 PhiC31 Integrase. Okay, an integrase is a type of enzyme that comes from a virus that is a recombinase. Okay, so recombinase does what type of molecular activity?
Recombination.
Recombination. Okay, so a recombinase is a specific enzyme that will do recombination. Okay, and PhiC31 integrase is from a virus. It does recombination at very specific sites. These are called ATTB and ATTP. Okay, these are just the names. So what this is PhiC31, say this is the protein. What that does is it takes these specific DNA sequences and then it causes recombination between ATTB and ATTP sites. These are just the names. This actually stands for ATT bacterial and ATT phage. I think that's what the P stands for. These are just little sequences of DNA that are a defined sequence that this thing recognizes and recombines. And once they recombine, the sites are destroyed, so it can never like recombine out. So it's a permanent recombination. So this is how we do Drosophila transgenesis nowadays. So to get this to work, and the reason it's site specific is because the recombination is happening at the ATTP. This is called the landing site. Okay, so the conundrum that researchers faced is, imagine this is one of the chromosomes of Drosophila. If you want to recombine something like a plasmid that has this ATTB with, say, your marker, you got your mini white, you got some gene X. Let's say you want to recombine a plasmid, and you're going to use PhiC31 to do that. What's the problem with the wild type D-mel chromosomes, which look like this? What's the problem?
Do they not have the site?
They don't have the ATTP site. So to solve this conundrum, you have to insert ATTP sites, okay, onto the Drosophila chromosomes. So what's the easiest way to get quick insertions of sites on chromosomes that I just talked about?
Reverse genetics.
Well, I wouldn't say reverse genetics, but the P element. Yeah. So what they did to get this to work, what they did is they built. So you got your helper. The helper just has the P transposase. You got your donor, and in your donor, you put an ATTP site between the inverted repeats. Okay. Then you microinject this. So when you're microinjecting this, what it's shotgunning into the genome is random sites of ATTPs, okay? So this is what they did. They built thousands and thousands, okay, so many, many, many lines of Drosophila that have sites, ATTP sites, shot into the chromosomes. Most of these are shot into chromosomes 2 and 3 in Drosophila, okay? And then they sequence all these lines to identify the site of insertion. And so there's libraries, or you might not call them libraries, but you might call them stock centers. So like Bloomington, which is at University of Indiana, maintains a stock center where they have thousands and thousands of Drosophila lines that have unique insertion sites for these landing sites, okay? And then all you need to do is you decide. The reason it's site specific is because you get to pick one of these lines. Let's say you decide I want to insert a transgene right between gene 1 and gene 2 on chromosome 2. So you have a very specific site you want to insert something into. All you need to do is look through the lines, find a line that has an ATTP landing site in the site that you want it, okay? And then you can get a recombination of this vector plasmid into that site using PhiC31 integrase, okay? So now let me just quick explain how they use the PhiC31 integrase, okay? So PhiC31 integrase, what type of molecule is that? Anytime you see A, is what type of molecule is that? What?
Enzyme?
It's an enzyme, so it's a what type of molecule?
Protein.
It's a protein, okay, it's a protein. So that means it has a gene that codes for PhiC31 integrase. So there'd be two ways to do this. One is if you wanted to get this integration, you could microinject. So if you could microinject an egg with the actual protein, PhiC31 integrase protein, plus the plasmid that had your ATTP site and your gene insert. You could microinject that. That's not how they do it. What they do is, so you have one fly, this is how they actually do it. You have one fly that's transgenic for ATTP. So its genotype is ATTP over ATTP on some chromosome. Let's say it's chromosome 2, okay? So that's coding for this. They take another fly that's transgenic for PhiC31 integrase. PhiC31 over PhiC31. And the construct that they actually inserted for PhiC31 would look like this. So somewhere on that Drosophila chromosome is an insert of PhiC31 integrase with a very special promoter. What kind of promoter would you want to get transgenic insertions, site-specific transgenic insertions in Drosophila embryos? What would be a good promoter? Or a bad promoter? If you give me a bad promoter, I can tell you why it's wrong, and then we can get closer to a good promoter. Take a guess. What kind of promoter would be good for this?
Myosin one, or always on.
That's a clever, okay, so that's a clever idea, is you'd want, in theory, you want a constitutive, high-powered promoter. Okay, so so they probably would have originally tried that right away. The reason that did not work very well is because many things that are classically constitutive are actually not expressed in early embryos. Because most from Drosophila development, most of the early expression in Drosophila embryos comes from where? This is kind of like the whole point of the last lecture, is most of the messenger RNA that gets loaded into the egg comes from where?
The mother.
Yeah, the nurse cells, the ovaries. So when Drosophila embryos are developing, the first like 1 hour, they're really not even transcribing stuff. They're relying on the messenger RNA that came from the mother's ovaries. So what would actually be the better promoter instead of a classical constitutive promoter?
Like one based on like some type of sugar probably, because they're gonna be laid on food, right? The eggs.
Not, well, okay, so sugar is not, so sugar is a,
Well, like galactase.
you're saying like a well, okay, so that's an enzymatic reaction. So is the early embryo gonna be expressing enzymes to get energy?
No.
No, because it's loaded with yolk protein, which it's consuming. So so the eggs don't eat anything. And the eggs don't classically like metabolize stuff. They're metabolizing the yolk protein that's already been deposited. So decent thought, but not a metabolism enzyme. What would be a better promoter?
Inducible?
The problem with inducing is it's very difficult to induce inside the early embryo. So again, like this should not be hard in the sense of just think back of where the messenger RNA comes from. Where does the messenger RNA comes from in the early embryo? It comes from the mother's ovaries, and those ovaries encode genes that are controlled by what types of promoters? Okay, so maybe I'll just also define there are what are called tissue-specific promoters. So a tissue-specific promoter, so like there's eye promoters that only express in eye cells. There's brain promoters that only express in neurons. So what would be a good tissue-specific promoter that would be good for this cause?
Ovarian.
Yes, okay, ovary promoters. Ovary promoters, or classically what you actually call this is maternal effect promoters. Okay, so there's maternal effect genes that have maternal effect promoters that are expressed in the ovaries. So most of the, to get early expression in the early embryo, you typically express it in the mother. And so if you put something like nanos, which is a maternal effect promoter, in front of the PhiC31, that's what this mother has. So it's a PhiC31 insert with the ovary promoter. So that's kind of like a key insight that actually is pretty important. Okay. So then, again, we're getting to how this works. You take a female that has a homozygous insertion for PhiC31 integrase regulated by an ovarian promoter. You cross that female to a male that has a homozygous ATTP landing site. Okay. The genotype of the F1s, so this is F0. The genotype of the F1 then is going to be PhiC31 over ATTP. So now you've got a fly egg that has both the integrase expressed from the mother in the embryo and the landing site. And then these are what you microinject with your plasmid that you want to insert. Okay? And then once you microinject these, this is already active. It grabs the plasmid and pops it into the insertion site. Does that make sense? That's actually like really complicated Drosophila biology, but it's fairly simple to understand, I think. Maybe? Any questions about that? How much time do we have left?
9 minutes.
Now, okay, good. Okay, that's perfect. Okay, now it's going to get even more complicated, genetics. But this is like my favorite stuff because like this is what we do. Okay. Okay, so so that solved the problem of now you can get site-specific insertion of any transgene on any chromosome anywhere you want in Drosophila, pretty much. Okay, so this is a incredibly robust system, and companies just do it. So you just build the insertion plasmid, you send it to a company, they'll do all this, they'll send you back a transgenic fly. But it's important to understand how it works. Okay, so now we're going to talk about the another really cool, complicated genetic system in Drosophila called Gal4 UAS. So this is what's called a binary expression system. Okay, this is one of the tools that makes Drosophila transgenics so powerful. So the purpose of building a system like this is you want to be able to engineer tissue-specific expression. So remember as we're talking about transgenics, we talked about in the past, like you might want to insert a gene that gets expressed in the chloroplast, or you might want to insert a gene that gets expressed in different regions. In Drosophila, we have such advanced systems that we can take any transgene and we can express it in any tissue whenever we want. So you could have some gene that you want to express in an eye, or in a brain cell, or in a kidney. They don't have kidneys, but they have malpighian tubules, or in a gut cell, etc. We have that kind of specific modulatable control with transgenes in Drosophila. And the way that it works is this Gal4 UAS system. Okay, so Gal4 is a transcription factor. Okay, it's originally from yeast, and so they inserted it into Drosophila. The Gal4 encodes for a messenger RNA, which gets translated into a protein called Gal4. And the Gal4 protein is an enhancer that turns on genes that are upstream of a sequence called UAS. So the UAS sequence stands for upstream activating sequence. Okay? And if you put a UAS sequence in front of some gene X, Gal4 can bind to the UAS and it can turn this gene on. Okay? So what we can do in Drosophila is if you want to get tissue-specific expression of gene X, which part of this double system, so these are actually two separate transgene insertions, which part of this transgenic system that I have not drawn yet do you need to modify to get tissue-specific expression of gene X? What would you modify? Anybody have any ideas?
And you haven't drawn it yet?
I haven't drawn it yet. There's one thing we can add, we can pop in, that will give us tissue-specific expression of gene X. It's over here. It goes right here. Yes. Okay. So let's say for example, we want gut specific expression of gene X. We want gene X to be expressed in gut cells. Okay. All you need to do is have a promoter, a gut promoter in front of the Gal4. Okay. And what that's going to do is, if there's a gut promoter in front of Gal4, that means in the gut cells, Gal4 will get made. Gal4 will bind to the UAS and it will turn on gene X. Okay, so do we all understand that tissue-specific expression part of this? Okay. So then you might ask the question, or you might be thinking, well, why not just do gut promoter gene X? Why not just do that? The beauty of, like, this would do the same thing. If you design a construct that was this, it would do the exact same thing. The beauty of this system is, sorry, let me go back. Why would you not do that? So typically, this sounds nice in theory, but in reality, what happens in science is you try to build this construct, and you insert it. What's the most likely outcome after insertion?
It drops it.
I wouldn't say that specifically, but I'd say actually the most likely outcome in most scientific experiments is it fails. It just doesn't work. Like, that's actually in reality the most likely outcome of doing this insertion. Something will go wrong, it won't work. Okay? So you really want systems that you can quickly optimize and try a bunch of different stuff, so that you can empirically find the best gut promoter. So if you just do this insertion, you're stuck with this. That's the genotype that you're stuck with. And you'd have to build a whole nother construct, do a whole nother series of microinjections and collect a whole nother series of lines to test a bunch of different promoters. But if you build this, if instead you just insert this system is much more easy to test quickly and optimize, because you don't have to build anything else. You build this once, and then all you'd have to do is buy a bunch of lines that have different Gal4 promoters. So you'd go to the stock center. And let's say they have a suite of hundreds or thousands of Gal4 lines that have different promoters. So there's probably 100 different gut promoters, promoted Gal4s that somebody built long ago that have now stuck in the stock center. You could just buy these. So you could say, okay, I'm gonna buy 5 of these. I'll buy 1, 2, 3, 4, 5 different versions of Gal4 with 5 different promoters. I'll build this construct, and then I cross these two together. And as soon as you cross them together, it activates the gene X. And then you can just quickly empirically test which one of these is working the best. And if it's that one, then you do your experiments with that one. Does that make sense?
And those are live flies that you're buying?
These are live flies that are maintained in a stock center warehouse, where they're all in these little tubes, and they're all data-based, and you can find them on an online library database and then buy them, and they send them to you. So again, just to wrap it up quick, the reason that this works is you have one fly that is your UAS gene X. And in this case, the gene is off. In this fly, the gene X is off. That's another reason the system is good, is because most of the time, you want kind of inducible control over your gene X. If you were to build your gut promoter gene X, that's not inducible, okay? But if you build this, its native state is off, and then you can get your fly line. Let's say it's Gal4 with some promoter 1 over promoter 1, Gal4. Then you cross these flies, and then the F1 generation is promoter 1, Gal4, UAS, gene X. And now in this fly, you've turned on gene X in whatever promoter tissue-specific promoter is regulating Gal4. Okay? That's kind of like really fast overview of Gal4 UAS. But are there any questions? Does that make sense?
Do you have to be heterozygous for the work?
Say that again?
Do you have to be heterozygous for the work?
I can't understand what you're saying. Do they have to be heterozygous for
Yeah.
what?
The system to work, yeah.
Oh, in this case.
Yeah.
So there are pluses and minuses to being heterozygous. In this case, they're heterozygous for both genes. If they're, you can make flies that with genotypes that look like this, that are homozygous Gal4 and homozygous gene X. This would still do the same thing. And actually you'd get double expression. You'd get more powerful expression. So they don't have to be heterozygous, but it's convenient that you can just simply do a simple cross and the heterozygous will turn on the transgenes. You can generate the homozygous, but it's through a process of kind of like more complicated Drosophila genetics. That takes a little bit of time to optimize. Any other questions? Okay, cool. That's Drosophila transgenics.